Hi, Dan Guerra here from VerEvMed Studios in the inland Pacific Northwest of the United States. Not far, but far enough from the Canadian border. Been away, now I'm back, and today we're going to do a two-part stunning video lecture series on immunosynaptogenics as well as epigenetic patterning of the central nervous system. And we're going to, at the end of the second lecture, I'm going to split this in half, actually derive a new theory, which I've been working on for some time. So you get a bonus today. All right, so let's just get started rather than wax wonderful about what you're going to see. Let's just do it, right? Of course. All right. So the topic today, as I said, for part one, is immunosynaptogenics as a mechanism for brain development and function. This is VerEvMed, of which I'm a co-founder and the chief science officer. That's me, Dr. Dan Guerra. There's my email down at the bottom and my Facebook page where you can find all of these video lectures. The background there, of course, is good old area around Moab, Utah, with the LaSalle Mountains in the backdrop. Lovely place. Probably need to retire there sometime. All right. Sometime in the distant future. Let's get started. All right. Now, this paper comes from Nature Communications. It's one of those Nature publications that actually has some open access. Nature is usually very uptight and puts behind all of their articles behind a very rigorous and stalwart paywall. But I think Nature Communications has a lot of open access. At any rate, this paper was published online 26th of March. Today is actually the 11th of April, so not that long ago. And I don't believe it'll be out actually in print press until probably the April or May or even June issue. All right, so what's this paper about? Well, let's give it a little bit of background like I'd like to do so you get kind of like comfortable into what we're going to talk about. So microglia. Now, a lot of the things I'm going to talk about today probably need definitions, but microglia are basically resident macrophage in the central nervous system. What are macrophages? They're part of our innate immunity. Macrophages, like the name means, is phagos means to eat. So macrophage, large cells that eat other things. What do they eat? Well, things like cellular debris, even whole bacteria and viruses, right? So macrophages their business is to consume and digest material that's left behind, maybe some killing spree carried out by the immune system. They also are active players in that process of detecting, notifying, and assimilating all the armamentarium to kill things. But their macrophages in general are just considered circulating cells usually that go around and digest all the material so it doesn't collect, and that material collecting up could end up being a foreign antigen, right? With the wrong kind of cross-linking and other kinds of modifications of proteins, lipids, and nucleic acids that could lead to the immune system to be constantly turned on and maybe generate an inflammatory response. So microglia are those macrophages they're an unusual macrophage, but they're found in the central nervous system. So think of them as being in the brain. There's a lot of these. There's probably more microglia than there are neurons in the brain. And they have a lot of functions in the brain. We'll get into that. But they traffic in the uninjured central nervous system, where they extend and retract processes. Those are cellular processes via actin machinery, actually, through the extracellular space, contributing to synaptic maturation and function. It's been known that microglia help in synapse formation, maturation, and also, as we're going to find out today, a constant rewiring, an event ontology of synaptogenesis, hence the title of the talk. So we know that early postnatal microglial morphological maturation, so the microglia themselves, correlates to synaptogenesis. That is the genesis of new synapses. And that's where all of the neurotransmitters have to cross over that sea of the synapse to go from the pre to the post-synaptic through the synaptic cleft to generate action potentials at the postsynaptic neuron in order to carry out nerve transmission. Right? You should know this. If not, you're getting a very short primer of it right now. At any rate, they correlate to synaptogenesis via neuronal signaling. So the microglia chemically signal with and physically signal with the neurons that they're associated with. And they do it through membrane-localized protein receptor reorganization. Okay, topic that we talk quite a bit about at VerEvMed, various reasons, a lot of disease states, for example, including CNS disease states. Microglia likely differentially process synapses during development. So when we think about developmental patterning, as the brain starts to pull together its network, right, where all the synapses fire and wire together, and you start getting an actual three-dimensional structure that is the brain that carries out the processes we relate to this called the mind. It's constant learning organ, the brain is. So that means you need to have some kind of event ontology to be able to constantly retailor how neural transmission is going down, right? How it's happening in real time. And rather than the neurons constantly changing chemically or physically, it's the microglia that help the neurons carry that out. And it's mostly at the synapse, the area between the two neurons. Okay. So here's a whole lot of information. I know some of you love this, and some of you, walk away and, do something while I'm saying all of these words. But it's necessary for me to get this out here. If you were in my studio, I would be able to use the chalkboard, but I haven't quite set my camera up for that yet. Maybe someday when I get a good studio assistant, which I know will happen any week now. All right, so let's take a look at this. Knockout mice lacking complement. Now, complement is a protein-based immunoreactive system that deals with removal of antigens. Many of you know this. Those that you don't, that's adequate for this short bullet on this slide. Show cortical excitatory hyperconnectivity, suggesting that microglia remove unnecessary neocortical synapses. Now, if you've heard several of the last talks I've done about the central nervous system, when we're talking about drugs such as cannabis, such as ethanol, such as cocaine, such as heroin, you'll remember that a lot of what goes on at the synapse are discussions of things like dendrites. And we know that there's a lot of, there are processes in that synapse that need to be pruned back, okay? Now, the processes themselves generate what's known as neuroplasticity, but neuroplasticity can become very confused or blurred if there's too much of that cellular process between the synapses. That's to be pruned back. So the microglia do that. Now, saying a complement's involved in that, and if it's not there, you get hyperconnectivity, too much connection between the pre and postsynaptic neuron. That means that there's a lot of extra noise, and that's what happens during early stages of development. That is post-parturition all the way until about 2 or 3 years. There's a lot of neural pruning. And then between 2 years and about puberty, 13, 14, depending on sex, depending on the genetics, and then there's yet another massive retailoring of the neural system using microglia again, as we know, when a person's in their early 20s. That's why they say that most people you can't have a really rational conversation with someone until they're over 20, 21. I know that that's terribly argumentative and biased. And all I can say is that I have experience in this, and I'm not going to make any further comment on it. All right, so you get where we're at here. Now, failure to eliminate synapses at the correct developmental time, this is not getting serious, prevents the normal strengthening of synaptic connections. And that's when you can lead to a lot of neuropsychiatric disorders. In fact, some kinds of autism spectrum disorder are linked to that, as well as a lot of inborn errors in metabolism known as sphingomyelinases. Now, those particular diseases and other lipidoses, sphingolipidoses and other lipidoses, I've covered in other talks, and I refer you back to those. Now, microglia may be required for environment-induced brain plasticity. Now, that very simply, you have a genome, then you have the environment, and you have gene by environment interaction and that leads to an endophenotype, not only at the neural level, but at the metabolic level, the endocrine level, the whole organ level. Everything in the body responds to the environment through time, adjusts to that environment, and then sets forth a new set of conditions or state functions where that new cellular mass or entire organism is now changed because it has gone through an experience. The experience then translated at the chemical and physical level, at the cellular level, at the biochemical level. Okay, that's what we're talking about here. And brain plasticity, for me to talk about multiple times, that's the new gadfly byword for, oh, everything's great if you have a neuroplasticity. It means that your brain is learning all the time. Like every time you listen to one of these lectures, there's a lot of neuroplastic changes, I'm sure, more than what happens in a whole week of not listening to one of my lectures, right? Maybe even a whole month, maybe for a whole year for some people. Well, at any rate, I digress. But the point is, neuroplasticity is considered good. Now, you will remember that the overt or extra neuroplasticity deals with this condition where you get a blurring of neurotransmission. And this is what often has been linked. It's called arborization or neo-arborization, with what happens a lot of times with certain drugs that people are taking, such as tetrahydrocannabinol, ethanol, cocaine, heroin, which are some of the major drugs that people unfortunately consume. Also, aging can result in that, or this condition where microglia don't do their work. Okay, so they're essential for synapse formation. Okay, and learning-induced synapse formation has to constantly happen. So these researchers set out to test the hypothesis. If microglia remove synapses during this is a mouse model paper, hippocampal development, using confocal microscopy analysis of microglia synapse interaction in fixed hippocampal tissue. So this is going to be a cellular study using brain slices from the mouse. Okay, that's an important thing to get. This isn't a human study. Now, using correlative light and electron microscopy called CLEM, which you'll love that particular acronym, CLEM. I don't know who thought of that, probably a geneticist, although cell biologists are doing this work and they're almost as bad. Of course, I'm not biased. Anyways, using CLEM, focused I-beam scanning electron microscopy, okay, now it's FIB-SEM, there is evidence for the partial elimination, or what is known as, and I love this word, trogocytosis of presynaptic boutons and axons by microglia. What does that mean? Trogocytosis is kind of like phagocytosis, or that is engulfing a cell or engulfing a membrane and consuming it, right, via the phagolysosome, breaking it down into individual elemental components like amino acids and individual lipids and individual maybe nucleic acids and carbohydrates, and then using those spare parts to resynthesize macromolecular structures, for example. That's what phagocytosis means. Also, phagocytosis involves the total destruction, consumption of things like viruses and bacteria intracellularly. Well, trogocytosis is not full-blown phagocytosis. It's basically just removing a part of the membranal section and then consumption thereof. That's what trogocytosis is. So it appears that using this CLEM and this FIB-SEM technology, which is basically spectroscopy and microscopy combined, you end up seeing that you get a partial elimination or a trogocytosis of only the presynaptic boutons. Remember, that's the part of the presynapse that's actually involved in neural transmission, right? That's where all the activity is. It's where all the messenger RNA is translated into, for example, neuropeptide. Okay, so that's an important issue. Now, trogocytosis has been described in the immune system as a non-apoptotic mechanism for the rapid capture and membrane components, that differs from phagocytosis. Now, I've been already saying this. It involves the engulfment and elimination of larger cellular structures. Okay, now that's what phagocytosis does. I just told you trogocytosis is limit of that. Using time-lapse light sheet microscopy of microglia synapse interactions in organotypic hippocampal cultures, trogocytosis of exclusively presynaptic material was observed. Time-lapse microscopy also revealed a frequent induction of spine head filopodia. Don't worry about all of these unusual terms, but spine head filopodia are going to be part of the neural process, the cellular processes, at sites of microglia synaptic contact. Okay? And don't worry, I'll give you more definition of philopodia in a moment. In the paper we're going to look at, it suggested that evidence for the elimination of synaptic material by microglia, resident microglia, in living brain facilitates neural circuit maturation by a combination of trogocytosis, that's a mechanism, of the axonal compartment, and the remodeling of postsynaptic sites, usually membranes. Okay. So, first thing they did is they say, do microglia phagocytose? You just saw in their introduction it doesn't. And look, wow, it doesn't. Number of spines, those are neurite spines, in opposed versus encapsulated versus phagocytosed. So opposed just means you get microglia opposed or pushed next to the neuron. You get a lot of spines, right, in that interaction. Encapsulated, that is surrounding, you get less, but still a lot of spines there. Phagocytosed, zip, zero. There are no spines that are completely phagocytosed. That's why they're saying microglia don't phagocytose dendritic spines. Now, the presynaptic membrane is the substrate for the elimination by microglia. So what the data showed was that, and it confirmed what we already said, double membrane inclusion bodies within this microglia, and that suggests that presynaptic material is a substrate for elimination. Let's look at this data. Okay, these are the, let me put my glasses on, love my glasses, number of inclusions, and here's the complete system showing you there's a lot of undetermined inclusions there. That means you're looking for entire synaptic digestion, so there's a lot of undetermined material. You can't figure out what there is, but when you look at the partial section of a synapse, now you start to see the floor demonstration of what's going on. There's no undetermined cells involved. There's a lot of microglia, okay, which is an aspect of microglial self-engulfment. There's a lot of axon, and there's a lot of the presynapse itself, which are the boutons, you see? So this is where the activity is, in that partial digestion, that trogocytosis, okay? All right, now let's continue with the data. This is data from the paper. The material being engulfed from boutons and axons typically range from 0.01 and 0.05 cubic microns, with an average diameter of about 253 plus or minus 24 nanometer. That is showing you here is 0.1 to 0.5. You're starting to see the microglial and axonal show up. Right over here, it's all completely undetermined. And as you move from the 0.05 to 0.1, you lose now all the axonal presynaptic, and now you're just back to 100% microglial engulfment. So the larger the size of the inclusion, you move away from where all the activity is. So all the activity for trogocytosis is right here in these very small diameter material. That's why it's only a partial section of the synapse, of that presynaptic membrane, very small diameter where all the activity is. That's what this data is showing you. Though this shows, I wonder why that says that presynaptic structures are not entirely phagocytosed by microglia, but rather they're trogocytosed. Love that conjugation. A term originally coined, here we go, to describe membrane transfer in immune cells. So trogocytosis used to be talking about chunks of membrane being moved from one immune cell to another. That's where the term was originally developed in the scientific literature. All right. So the presynaptic bouton or axon material digestion by microglia looks something like this. Right? The microglia is in the red and the neuron or the synapse is in the blue. You get invagination, you get sinking, you get closure around that synaptic area, then you get trafficking and movement, and finally degradation. This is the trogocytosis of a portion of the membrane of the presynaptic system. So boutons or axonal pinches, that's what this is here, are engulfed into the microglial cytoplasm. This is just red-pink color here, before closure and membrane and subsequent trafficking and then degradation. This suggests, this mechanism suggests specific trogocytosis of the presynaptic structure by the microglia. Thus, trogocytosis is directed toward axons and synaptic boutons, as we've been saying, rather than selectively targeting the entire presynaptic active zone. It's very specific here. Okay. Now, more data. Microglia induce spine head filopodia formation. Love that spine head filopodia. Yeah, I tell you, I just love the terminology that you learn when reading literature you don't normally read. Now, this is the number of spines retracting. And you can see this is the transient spine, and a fair amount of that is retracting. And there's persistent spine when the microglia is contacted with the synapse. But when you have non-contacted microglia and presynaptic structures, you have only the persistent spine that is retracting. That means that all the transient spine isn't retracting. That means something's happening to it. We already know what it is happening. It's being trogocytosed. Right. Okay, more data, you see. Now, let's read. Microglia spine contacts were non-random, very specific, as we said, certain axon regions and certain boutons of the presynapse. Transient spines were found exclusively among contacted spines, where the cells actually contacted one another, when compared with nearby non-contacted spines. See, 13% of them there, 0 in non-contacted regions. That means there's a chemical gradient that's being generated between the microglia that are doing the work and the presynaptic cleft where the work is being done. These spines formed from filopodia, and there's actually a filopodia forming there, where they put that little arrow. See that? Coming from the synapse, that filopodia, that it's called a process or an extension of the presynaptic membrane. And it appeared at the microglial contact point, and that's exactly where it's happening. There's the microglia coming down. You see the filopodia forming, that pinching off of that presynaptic membrane. See, isn't it beautiful photographs? See, that's what you do when you combine spectroscopy with microscopy. Nice stuff. And by the way, this paper has a lot of movies. So you should go check out this paper if you like to watch movies of subcellular interactions. Really cool stuff. I'm actually not a real fan of it, but I know a lot of people are. All right. I mean, of the movies. I love the paper, actually. Okay. So mature spines contacted by the microglia formed a filopodium protruding from the head and extending toward the microglial process. That's what you see here. Filopodia, and here's my definition. I went and looked it up for you to give you a more classical definition rather than the silly, sloppy way I was describing it. The filopodia are, listen to this now, drum roll, membranous, actin-rich protrusions, okay, that chemotactically, I've used that word already, probe their environment. I got that from Nature Reviews, Molecular Cell Biology, a 2008 paper, where they first started describing filopodia. Okay, so it's the same thing I've been saying. Now, actin, again, is a motor protein. So in order for these processes to move, to actually translate a position into three-dimensional space, you need motor proteins like actins to carry out that process mobility. And so they're actin-rich protrusions, okay? Chemotaxis are moving towards some chemical gradient towards that microglia. Okay. Filopodia that are involved in cell migration. Here's some things we need to know about them. Neurite outgrowth, which is kind of what we're looking at here. And they serve as precursors for dendritic spines and neurons. So these filopodia are really important, again, for neural system generation and constant retailoring. Okay, really important structures in the process. Here you are looking at them, right? Excellent. Scanning electron microscopy. All right. The initiation elongation of filopodia, shown there by the arrow, depends on precisely regulated polymerization, convergence, and cross-linking of those actin protein filaments. Okay, so that's the actual motor mechanism that moves that along. Microglia generate filopodia during neural synaptic spine formation. Now take a look. Look at the way these filopodia move. They're very much directed vectorially towards that microglia. So there's a vectorial direction. This is just showing you they could be moving isodiametrically anywhere around the 360 radius of where you find that synapse. But no, they're directed towards the microglia. So obviously that is the potentiation of activity towards the microglia. So that's a chemical gradient. And again, you're seeing the same thing here. The number of spines. Here's spine stretching, here's filopodia and spine head filopodia down here. And in the contacted region, you see a lot of spines doing that. That's when this protrusion is occurring. It's when this protrusion is occurring, and then the non-contacted very little. So once you contact, you start generating this philopodia. You start carrying on. Once the chemical gradient allows for philopodiogenesis, this is when you start getting all of this activity, and the number of spines starts to grow. Right? Again, this is important for understanding how you develop the synapses, which are constantly reorganized as you learn and as you forget and as your memory re-involves the past after you get a sleep cycle and you start to consolidate information into actual long-term memories. All of that is what is induced by this system you're looking at here in these micrographs, as well as the data you're seeing here. And there's a really beautiful picture there. Again, there's the microglia, right? This is done in red here. And then there's the philopodia in green. Okay. You can see multiple philopodia are extending toward the microglial process. Okay. And the way they got these different colorations is they used different levels of gene expression, okay, which fluoresce differently. So beautiful microscopy and stereochemistry as well as photography. All right. So what's the summary of this paper? So it's just going to be in two parts. So I'm not going to spend too much time on this paper. That's all we really need to look at for this point. What do I do when I want to summarize what we just looked at? I didn't show you all the data because most of the data is in movies and in a lot of those scanning sections. It's just not going to mean much. You saw how the data was summarized. Not going to mean much for this kind of talk. Okay. So what I want to do is summarize what it says. Microglia directly engulf and eliminate synaptic material. Eliminate, get rid of it. Okay. There's no evidence for phagocytosis of the entire synapse. That's good, because can you imagine removing entire synapses? You spend all this time before these synapses to learn the word trogocytosis. And then all of a sudden, you walk away, you walk outside, you take a walk out in the forest out there, and you forget what trogocytosis is. That's not good. That's what would happen if you had the entire synapse removed, right? Because you didn't need to have that understanding through that endoneural phenotypic change associated with the synaptic tailoring. But that's not what happens. You get a partial digestion or trogocytosis, so that not only do you remember it, but you remember it within the context of what I was saying in this lecture. See? So it allows for coherent activity of memory and real-time learning and understanding. Okay. And I'll develop that later when I give you my theory in part 2. Microglial trogocytosis was restricted to the presynaptic boutons and axons. Okay, that's what was shown, with no evidence for elimination of the postsynaptic material. So that's interesting. So it's the pre-synapse. It looks like it's becoming tailored at the molecular and cellular level. So that means that it's where the neurotransmission is coming from that gets tailored, and not where it's going to. And I can already give you a good sort of wavy gravy reason for that. Okay, hand waving, wavy gravy, depends on what kind of metaphor you want to use, right? I just saw that wavy gravy, and it does not bring good things to mind. So maybe we won't use wavy gravy. We'll call it hand waving. Okay, that hand waving thing about having this occur not at the postsynapse. Here you go. The postsynapse is a responding system, right? And it responds by stimulus, by recognition and stimulus, right? Acquisition and apprehension of the stimulus, and then further transduction, giving you the action potential to now the postsynaptic neuron. So they can now carry that action potential forward. So since it's on the receiving end, it's going to be only required to receive that which is being transmitted. So if you were tailoring both the pre and postsynaptic, you would have to have in real time a complete understanding, and again, that's a terrible metaphor, but a complete detection by the system right there of all the micro concentrations of all the neurotransmitters, the salt concentration, the pH gradients. That's way too much to be happening in real time. So the system works only on one side. It alters the presynaptic system, and then whatever makes it to the postsynaptic system is received, come, thank you, yes, arrive on our shores, right? And upon receiving that, all of the membrane receptors that are in the postsynaptic neuron are able to accommodate, and then they change over time depending on the signal stimulus. So that's your armchair or hand-waving understanding of this. And it's more than that. I think it's a pretty clear description. Hopefully you got that. Anyway, Microglial contacts at postsynaptic sites elicited transient philopodia, most of which originated from mature spawns. Okay, we saw that. So there is an interaction there, but it's an interaction associated with trying to recognize the change in the stimulus, which is what I just said. Microglia digest membrane synaptic material affecting synapse remodeling. It's basically what I just told you happens, explaining the diverse synaptic alterations during microglial activation. Now remember, the microglia themselves have to be activated. Remember, these are resident macrophages or immune cells doing this. Who would have thought they'd be doing this? This is like major heavy lifting. This is actually controlling nerve transmission, full stop, plus it's controlling the neural network, three-dimensional temporal state, double full stop. So in other words, the microglia are doing most of the heavy lifting, I guess, too, in a way, on all the aspects of how the brain goes through experiential learning, right? It's the immune system, right? That is what I've been looking at for years, ever since 2008, when I started doing neurobiology and neurobiochemistry. And look what's coming to fruition now in papers now published early in 2018. Well, there you go. Anyway, immune-mediated neural development via actin, filopodial vector migration, explains that differentiation. So you need to know that there was actin in there, okay? So that, biochemists, particularly, I would say scientists, particularly biochemists, we really want to know, okay, what's the mechanism here? This is all good that we can see this. The movies are nice and whatnot. The colors are really dramatic. Wow. But I'm a biochemist. I want to know what proteins or lipids or nucleic acids are involved. Well, at least they gave you the protein, and it's the right protein. It's actin. It's a motor protein, right? So its polymerization and relaxation actually will involve process movement. So all kind of fits together. All right. Now that leads me to a beginning of my theory. Now, what I want you to look at here is a trigonal planar system shown on the left, where you have within this would be the central individual, that is, say, an individual human being's brain. The brain is going to develop according to interactions with the genome, with the environment, and with a third player, which I'm going to call the immunoepigenome, for reasons you'll discover later when you watch lecture number 2. But you already know that immunoepigenome's got to mean there's something to do with immune system and something to do with epigenetics, which that means what? Covalent modification of histones and chromatin that affect gene expression that are not embedded directly within the sequence of the DNA. Okay, that's what epigenetics is, remember? All right, because we talked about it at length in previous talks. Now, I'm giving you these interactions because what I want you to already understand here is that these are not substances. So don't think of the genome as a substance. Think of it as an event. A genome doesn't mean anything, just bare nucleic acid, even with protein such as in chromatin, condensed chromatin, unless it's doing something. So the genome is carrying out some kind of process event. So these are event ontologies. The genome interacts with the system's core, which is the self, which is in this case, the brain, okay, the individual brain. That's why each brain is distinct. Likewise, the environment interacts with the genome at the level of the individual, the existing individual. The third component is an immunoepigenome, which basically tracks the interactions between the environment and genome, facilitates a translation of that interaction via epigenetic changes using the immune system. So all three of these are working together to pilot and reform the central nervous system according to your basic hereditary genetics, the environment in which that genetics is embedded, the facticity of that, and then the mechanism whereby that occurs, the transcendental mechanism, is the immunoepigenome. Now that's similar, okay? That system reminds me that when you're thinking about self and non-self, like the environment, you can compare that to symbiotic associations, such as talking about, let's say, bacteria in the environment, or plants in the environment versus animals. Let's take a look at this. This system here talks about symbiotic associations. At one end of the spectrum of parasitism, which is unilateral benefit. Who benefits from that kind of interaction? Okay, the degree of harm to the host from none to severe, the parasite does. Benefit to two species, only one, the parasite. Both, you'd be down at symbiosis, such as when bacteria like Rhys Obeah interact with certain leguminous plants or Fabaceae plants that generate root nodules so that the plant can fix atmospheric nitrogen gas, right? That would be a symbiotic relationship. Those rhizobial bacteria at the root interface forming nodules at the root within the soil layer is actually a symbiotic relationship with bacteria get nutrients from the plant, and they also get shielded from molecular oxygen because of a certain lectin that's generated that binds oxygen, because these are anaerobic in that nodule system. And then the plant gets fixed nitrogen. Boy, pretty good, right? Plant can't fix atmospheric nitrogen. Animals can't fix atmospheric nitrogen. Certain bacteria can because they have the right enzymatic machinery. All right, that's a pure symbiosis. So between parasitism and symbiosis, you have things like commensalism, where there's some degree of harm to the host, but it's mostly an interaction that's shelter and food, okay? Such as living in an environment where the organism is sheltered by another organism. Maybe it has to give up some of its freedom of movement, but it still is getting something, some provision or benefit from it. And mutualism, where it's truly a reciprocal benefit, where one organism or one system interacts with another system Venn diametrically, so that interaction is mutually good for both, right? So this is like an ecological ecosystem approach to understanding how hosts interact with their environment. So if you want to look at the self as the host, and even though the genome and the immunoepigenome are part of it, they are really external to the actual event ontology that occurs when the environment interacts. Now, let's take a look at this now in the square of opposition. So now I'm taking the square of opposition from categorical logic. Living systems interact according to a pseudo three-dimensional biological trigonal plane of opposition. This is something I'm proposing here. Okay, it's pseudo three-dimensional because it's a flat plane shown here, but the dimensions actually come out to not 3 but 4 because you have time. So the way that I'm describing it is a pseudo three-dimensional, but you understand it's got to be 4 dimensions, right? All the spatial dimensions, there are 3 of those. Temporal dimension, that means 4. Okay, let's take a look at this and put it into our square of opposition. You have the universal affirmative. No harm to the host. Maximum benefit to both. Rare or occasional dependence. Okay, entirely no dependence, right? No problem with problems to the host, and basically both have benefit, maximum benefit. Okay? That is like a symbiosis, okay? And some forms of commensalism. Now, the universal negation of this interaction would be the following. You follow me here. Severe harm to hosts, benefit to only one host, or say the parasite, 100% dependent. So you're 100% dependent on the system. The host here is not getting anything. It's severe harm to it. Okay, so the parasite's dependent on the host. And only one of the organisms, in this case, the parasite, let's say that's invaded the body. Let's say it's in helminth, for example, the most gross one I can think of right now, the most gross example, some helminth in your gut, right? Or in your liver. Ooh. That would be a parasitic host, right? It's not benefiting the host at all. It's benefiting itself, and it's totally dependent on that host, right? Okay. Then there is, okay, so that's severe negation. Then there's particular affirmative. Some harm to both. Benefit is 50%. Dependence is disinterested, meaning it's impartial, okay? So that would be a particular affirmative. That'd be like somewhere in the middle of each of these arrows, somewhere in the middle, somewhere where you get the Venn diagrams all come together. A little bit commensal, a little bit mutualistic, a little bit parasitic. Now, the fourth one is the hardest to get your mind to wrap around. It took me, I bet, 5 minutes to put this one together, but I think you'll get it if I correctly describe it. Particular negation, which again is that fourth corner, the lower right-hand corner of the square of opposition. Some harm to neither. Okay, so that's a weird way of putting it. It doesn't mean no harm. It means there's some harm to neither. That means you can't really describe harm to either one. Okay, doesn't mean it's not there, but you can't really describe it. So some harm, benefit to neither, doesn't seem to be anything really working here. So it's like there are two, let's say, organisms in an ecological niche, but doesn't seem to be enough interaction to benefit. And then because there's no benefit, there's no dependence. Okay, so that would be particular negation. Now, if you recall when we talked about the square of opposition, for example, in aging and other times when I've harnessed logical principles to discuss biological, biochemical phenomena, you'll remember that almost all the time we end up saying that things are either particular affirmative, particular negative. They're never universally affirmative or universally negative, okay? Because life does not work on those extreme universals, because you know that universals are nominal, or at best terminal, meaning not terminal like they're deadly, although in some ways I guess they are in terms of intellectualism, but universal meaning like it's only a term, right? So example is you have the universal term humanity. Okay, go outside and find me humanity. You could say, well, there's Joe and Sally and Pete and Rich. Well, that's not humanity. That's those four people. Those four people may possess something you call humanity, but it's not humanity. Even the whole mass of the entire human race, where is it? How can you locate it, right? So what we say as nominalists, we say universalism doesn't really make any sense in the real universe. It's just a term, right? It's a useful term. It's easy to understand and integrate into our logical understandings, but it's not a real thing that you can go out and look at as phenomenon, right? So it's not a real term, or it's nominal, meaning it's a name only. Okay. Now, we're going to skip this. This just has to do, it's an abstract that came out just this week that's talking about our friend Rhys Obeah and how Rhys Obeah interacts with the leguminous host in order to be able to make those nodules. I put that there so you can go slowly through this slide collection here, and you can read this, and you can go and look at science signaling. It's just the abstract, and see that people are still studying how that interaction works. I just put it in there for hoots and hollers. All right. Now, the interplay of organisms is the macrocosm, but it also appropriately describes the human body. So this is getting back to my discussion here of what we just looked at. And overall stress imposed by the microbiome, invading pathogens, autoimmunity, cancer, autophagocytosis, everything we've been talking about at VerEvMed for the last year. So the interplay of organisms is the macrocosm, but it also appropriately describes the human body, or the microcosm. And that includes the overall stress imposed by all of those things. Okay, thinking about the ecological grid, thinking about the square of opposition, and thinking about my trigonal planar description about those event horizons. So development, differentiation, and the signal transduction cascade, those are all parts of what? All parts of the genome, the hardwiring of the cell. That cascade network, including neuronal action potentials, endocrine mediation, endocrine hormones, for example, paracrine hormones, the whole nested set of signaling compounds, also compose an opposing three-dimensional trigonal plane, where the central element, or that is event, is the homeostasis. You see, that's an event. A homeostasis is an event. It's not a substance of the existing individual. So now I've taken cellular biochemical physiological terms and I've embedded them in my theories. Indeed, learning and the accumulation of memories and knowledge, which are justified true beliefs, are all part of a massive internal interactome that can be understood relative to advantage, vectorial control that we just saw with the microglia processes, right? So there's a direction to the tunneling of information to generate memory, ideation, and understanding, right? It's not just a rhapsody of sounds or sensations. There's a tunneling effect, right? There is a natural native system that encompasses all those features to be able to carry this out. And that indeed is the immune system. Now you're going to find when I talk on my second lecture that I'm going to want to rename the immune system. The immune system, people think defense, the immunity, right? Well, if we keep using that term, people are going to get more and more confused, especially newer generations of biologists. And lay people too increasingly want to be know have a good knowledge about the immune system because of all the drugs that are used, pharmaceutical drugs that are used to be able to control immunity. Okay, so I'm going to rename it. I'm going to give it a new name, but not now, later. All right. So I'm going to continue on with this dialectical discussion. Thus, the immune system has two roles in the human body. One is for defense, the immune system, and the other, in conjunction with epigenetic mechanisms, generates the existing individual with an ongoing neural network that is one that's constantly perceiving and acting upon and assenting, that can learn via attention and assent, Stoic terms, to stress on the system. This is accomplished via homologous recombination, okay, get back to the molecular level, remember we're doing biochemistry here, of variable regions of both the immunoglobulin family from B cells, plasma cells, and the T cell receptor also is reformed by recombination, in concert with chromatin remodeling, how genes get expressed, right? All the opening of the DNA, the opening of the double-stranded DNA, all the transacting factors that allow RNA polymerase to enter, all of the possible patterning that goes on in chromatin to remodel it so that genes that get expressed or recombined, plus the histone code, which is associated with the chromatin, and both the acetylome and methylome of cohering DNA. Acetylome is all the acetate that's loaded on there. Remember when we talked about acetate adding and removing on histones, on lysine residues histones, and on DNA, and of course methylation patterns on cytosine residues, and also methylation indeed on proteins cohering with the DNA. So that's all part of the system, okay? So if there is a link between the mind and the body, at least one component is physical. Absolutely. This is where René Descartes ran into a little bit of a problem, right, with the pineal gland. This connection might be the molecular. This is now, again, my proposal. This connection might be the molecular and cellular adaptive immunological interactome that serves to generate neural tracts, which we just now saw a glimpse of, according to developmental, okay, normal genomic development, endocrine, hormonal influence, stress, diet, etc., and the peripheral stimuli, which is inducing effects on the system, while maintaining repair processes in the central nervous system, because the microglia also do that. The immune system also does repair, right? By using the complex interactions between microglia and the neurons. Now, later on, you're going to find it's not just microglia, it's all the different immune cells. But right now, that's good enough. It was established that neuroinflammatory mediators play a critical role in the pathophysiology of brain ischemia. We know this. Anybody that works in a emergency room knows that brain ischemia, the first thing you have to do is stop the bleeding because you're getting a lot of neuroinflammation. You also have to get plenty of oxygen in the brain, or you lose the mind. You lose components of the brain, you lose the mind, sometimes irrevocably. So that's exerting either deleterious effects, like when this ischemia gets too bad, like a place where you cannot stop the anoxygenic system to function. Anyways, deleterious effects on the progression of tissue damage, such as necrosis, or beneficial roles during recovery and repair. So neuroinflammation does both. So it's already there. Remember, the innate immune system has the innate and the adaptive. The innate is non-specific, first line of defense. There are cellular and humoral, just means fluid components. The adaptive is specific. It's a second line of defense. It's protective, and it's also associated with exposure. And there are cellular and humoral components there. So let's take a look more at what we mean. Okay, this entire system comes from bone marrow. You have lymphoid stem cells. You have two different branches here. You have the myeloid stem cells, which form things like erythrocytes, platelets, neutrophils, which are your policing cells that move through the body looking for foreign antigens, macrophages, which consume material that's been digested. And then you have all this lymphoid stem cells. So that's the myeloid stem. The lymphoid cells, you get B cell formation. Okay, those are the cells that eventually will form plasma cells, which will make antibodies. That is immunoglobulins, those glycoproteins that get secreted through the plasma membrane via the ER and Golgi apparatus, which we talked about previously. And you also get the best cells in the whole system, the pre-T cell, or T cells in general, which mature in the thymus, or if they stay out of the thymus, they just become natural killer cells. Love those too. Seek and destroy anything that doesn't look right. But if they mature in the thymus, they generate epitopes on the surface of these cells called CD4 plus, CD8 plus, or double positives. Those can generate into Th1 or Th2 cells. Those are the T helper cells, or to the cytotoxic T lymphocytes, which do similar things in natural killer cells, but they're much more specific. So you have these two major lineages, right? All the myeloid lineage and all the lymphoid lineage. Now, microglia, what we talked about just now, just this one cell, which is just a macrophage subdivision. Look what it's involved in. It's involved in recognition, regulation, phagocytosis. Now we can add to that trogocytosis, and output. Things like what? For example, recognition. They can recognize the A beta protein, like in what? Alzheimer's disease, through this receptor system. Through RAGE, they can likewise, we talked about this in previous talks, about the A beta and age. These are motifs in proteins that can be recognized by the microglia. Here you got cytokine, chemokine receptors, right? Here you've got CD14, toll-like receptor 4, dealing with heat shock proteins or apoptotic cells. Here you have RNA, foreign RNA recognized by toll-like receptor 3, more A beta and toll receptor 2, the purogenic receptors, recognizing all these nucleotides, these purine nucleotides. Phagocytosis through all these various receptors, the mannose, the FC receptor, scavenger receptors. We talked about these before, like CD36. Regulation through all of these subcellular surface membrane components interacting with receptors involving things like chemokines and cytokines, right? Semaphorin receptor dealing with semaphorin 3A, something we talked about previously. Neurotransmitters and neuropeptides, all of those systems are going to be impacted by activity of the microglia. And ultimately, neurotoxic and pro-inflammatory. Hydrogen peroxide, nitric oxide, superoxide, interleukin-1 beta. These are pro-inflammatory interleukin cytokines, IL-6 and TNF, tumor necrosis factor alpha, and on and on. Okay, one cell carries out phagocytosis, recognition, output, and regulation. Remember that all those cells I just showed you have this same potential. Okay, so regulation of microglia in the CNS. Let's go back and remember what we talked about. Microglia and macrophages are activated following the stimulation of various recognition or phagocytic receptors. The activation state is subsequently controlled by neurons, which secrete or express numerous regulatory ligands. The end result of those interactions is the release of cytokines by the microglia, neurotoxic compounds, and or growth factors by the microglia, or the activation of cellular pathways, including things like trogocytosis or phagocytosis. Aberrant function of those pathways can result in significant degeneration during aging or disease. So you know that they're ultimately going to be involved in that too, senescence, for example. All right, so I know that there was a lot of material, and I know I went through a lot of high cognitive learning at the end where I was trying to introduce a new theory. Don't worry. Part 2, I'm going to go back to the literature. We're going to go through a paper and go through an epigenetic paper, another exquisite paper I found in literature, brand new. And then I'm going to bring back the theory that we were just talking about the immunoepigenome. We're going to rename the system, and we're going to give you more detail about how I think it might work and how we might have future plans to study it. So I'm going to end the talk now. That's me, Dan Guerra. That's VerEvMed, our company. There's our email address. There's our website. Contact me also at my email address, and my Facebook page is on the very first slide that you looked at. All right, so let's get this out of the way, and let's do like we always do. This is part 1. You'll see part 1 and part 2 together. I'll put them both up. I'll post them both on YouTube, hopefully at the same time, probably sometime tonight or tomorrow. That'd be late the 11th of April, 2018, or early the 12th of April, 2018. So until then, bye for now.